In lesson 2, we put nucleotides together, sugar, base, phosphate. In lesson 3, we explored the structure of DNA and what the importance of the 5' and the 3' end were, complementary and antiparallel. This lesson is going to serve as an introduction to both replication of double-stranded DNA and transcription of double-stranded DNA. The purpose of this is to show you, when you draw them correctly, how similar they are. And if you understand one, particularly replication, because of its advanced complexities we'll get into in the next lesson, if you can do one, you can do the other. And the concept remains very similar. Really, this is to give you some introduction to vocabulary and to prove to you how important 5' left, 3' right of the DNA you care about is. The DNA you care about is going to be specific to each lesson, but almost always the 5' left is the strand that's being made. Show you in DNA. Right? DNA is double-stranded, and when DNA is going to be replicated, it is going to be unzipped. Let's not worry about the details of the unzipping just yet. This black strand is the template strand. It has got a helical structure and another strand which is complementary and antiparallel to it. Haven't put up any laterality yet. No 3', 5'. The DNA polymerase, the thing that is going to copy the template strand, is going to start on its own strand's 5' end. And it is going to grow the strand in the 5' to 3' direction. Now the image that I've drawn makes it look like we've got DNA polymerase sitting at the same place and pushing out the DNA strand from the 5' where it started to the 3' where it's going. I think of it like that because it helps me keep things straight. 5' left, 3' right. The strand that's being grown, the replicating strand, the one that's in process, starts 5' left of the board, 3' right of the board. DNA polymerase starts at the 5' end of the growing strand, if it's the thing that it's making. This is where people get confused because we say DNA polymerase builds 5' to 3' but reads 3' to 5'. Builds, reads, what is that? This is why I say to do this. Start DNA polymerase on the 5' end of its own strand. Have the growing strand be what we've always said it to be. It must be the 3' end. The 3' is to the right. Because we're going to make complementary and antiparallel strands, it has to be that way. The DNA replicating strand, which is 5' left, 3' right, growing in the 3' right direction, adding to the 3' end, must be complementary and antiparallel to the template strand. All that means is DNA polymerase starts on its own 5' end and moves to the right. 3' end. The template strand is backwards, so the template strand must be 3' left, 5' right. But you orient the strand that matters, that one that's being copied, 5' left, 3' right. Now, DNA replication throws in an extra layer of complexity. The template strand is the one being copied. The replicating strand is the one that's in process of being made. In DNA replication of double-stranded DNA, both sides of the original DNA act as template. And at the same time, you've got another DNA polymerase that starts at its own 5' end and grows in the 3' direction, adding on to its own 3' end. Now, the original template strands were complementary and antiparallel to each other, which means that template 2 is going to be 5' on your left, 3' to your right. The mirror of the replicating strand, identical to the replicating strand. it has its own replicating strand, which is complementary and antiparallel. So if you ignore template 2 and replicating 2, you simply just don't see that they exist. What you'll get is a very simple picture of DNA polymerase starting 5' left, going to its own 3' right, when against the template strand, which is 3' left, 5' right, complementary and antiparallel, knowing that this other thing has to happen at the same time. We'll get into that a little bit. The reason why I want to start with this is because what I want to do is compare that to what happens in transcription. The top strand is the template strand. It's going to have an original, complementary, and antiparallel DNA. Only in replication, we had both DNA polymerases going at once, simultaneously in opposite directions. In transcription, you only have one enzyme. And so what I do is I place the 5' end of the growing strand to the left. RNA polymerase binds to the DNA at the place where it's going to start at its own 5' end, and it's going to grow the strand to the right in the 3' direction. Okay, that looks a lot like the top strand of the replication. And yes, indeed, that is why I do it this way. The 3' end to the right of the RNA transcribing strand looks an awful lot like the 3' end of the replicating strand. It's all about orientation. The strand we care about is the one being made. In DNA, we put the DNA polymerase on the 5' moving to the 3', 5' left, 3' right. In RNA transcription, we put the RNA polymerase 5' left, moving 3' right. They look the same, but the color changed. Now, in reality, what happens with the polymerases is they start at their own strand's 5', and the thing itself actually moves down the template strand. That's really what's meant by read 3', 5'. The way I've drawn it looks like the RNA polymerase is just kicking this stuff out, moving it to the right. When actually, really, the whole RNA polymerase molecule moves down the template strand, pooping out the transcription strand off the side. The tough part of DNA replication was that 2 templates are going at once. The tough part about transcription is what about that other strand? Right? We know the template strand is going to be 3' left, 5' right, because the transcribing strand we've oriented 5' left, 3' right. But since the original DNA was complementary and antiparallel to one another, and the transcribing strand is complementary and antiparallel to the template strand, the transcribing strand is going to look exactly like the coding strand. In this example, the black line is the template, complementary and antiparallel to the red strand, which is growing. The black strand is complementary and antiparallel to the white strand, which means that in sequential order, 5' left, 3' right, the coding strand is going to look just like the transcribing strand, such that except for the change of uracil for thymine, you are going to get exactly the same pattern of nucleotides, left to right. Now, very rarely do I mention the coding strand, because all we care about is the one that's being grown. What I want you to see is that they can use this in 2 ways. One, they can give you a template sequence, and you have to orient it 3' left, 5' right, so that you can figure out what the DNA replicating or transcribing strand is going to be, 5' left, 3' right, and make them complementary. Or they can give you the information of the coding strand 5' left, 3' right, which is in that sequence what the transcribing replicating strand is going to show. We're going to get into a lot of detail on this in the next couple of lessons. So this is meant to be only, it's important to put your thing that's growing 5' left. And I want to introduce some vocabulary and show you a little bit of the complexity. When we talk about DNA replication, people say that it is semi-conservative. What does semi-conservative mean? It implies sort of conservative, but no, semi-conservative means literally 1/2. That is, the parent DNA has 100% DNA. It's 100% black and blue, 50% black, 50% blue. Black and blue are complementary and anti-parallel to each other. When this process happens, when it unwinds, the blue strand is going to copy anti-parallel and complementary to itself. The black strand is going to copy anti-parallel and complementary to itself, which means that the two strands are going to end up, the daughter strands, an entirely intact black strand and an entirely intact blue strand. So 50% of the original molecule ends up in each of the children, semi-conservative. Which is weird to say because if you look at it, the black strand is going to make a complementary and anti-parallel strand to itself. The green strand is the blue strand. And the blue strand is going to make a complementary and anti-parallel strand to itself, the red strand. The red and the black are the same. It actually is identical genetic code if the copying goes right. They're equal to each other. But it's called semi-conservative to imply that the black and the blue of the original, the two strands separate from one another, copy themselves in a complementary and anti-parallel fashion so that the copies are the same, but each daughter has one entire strand, one entire half, one entire DNA string of the original. That is, the black and blue don't mix. The black goes to one, the blue goes to the other. The actual code that's copied is the same, but the original parent is separated into two. If we look at that concept under prokaryotic replication, we'll see the same thing, except their DNA is circular. Got a black and a blue. In order to copy both strands at the same time, they're going to have to unzip it, just like this top one. And what that's going to look like is a replication fork. The rest of the DNA is still the same, connected very close together, but at these points there's an enzyme that's headed off away from the bubble that's unzipping the DNA, allowing whatever the DNA polymerase happens to come by to copy it. Now the opening of this site is called the origin. If we zoom in on the origin, what we see is this. We have one strand, which is oriented 3' left, 5' right, and its complementary and anti-parallel strand, unzipping from it, oriented 5' left, 3' right. At the site of the origin is where the DNA polymerase is going to bind. It's going to start at the 5' end of its own strand and grow towards its 3' strand, which means the DNA polymerase is going to move towards the template's 5' end. At the top, this means going clockwise. On the bottom, it's going to be the exact opposite. It's going to bind the same origin site, but it's going to move 5' to 3', 5' right, 3' left, because the strands are complementary and anti-parallel to each other. Now, if it did this, what would happen is the entire thing would be copied in opposite directions, and everything towards the 3' template side on either side would be left untouched. Now, teleologically, that makes sense. You just unzip the whole thing and just start going. The problem is that when you have single nucleotides exposed, as this would be, behind the start of the DNA polymerase, if you had exposed single nucleotides, Bi doesn't like that, it would degrade them. And so rather than waiting for the DNA polymerase to go all the way around to catch up, what happens is you get multiple start sites. Not red to imply RNA, red because it's the only color I have. These short bursts of DNA polymerase are called lagging strands. Lagging because they are not built contiguously and will eventually need to be connected together. Whereas the leading strands are going to go and be continuous and stop it only when they encounter one of these lagging strands. And then if we look at it under eukaryotic situation, the same thing is going to happen with a little more complexity. The eukaryotes do not only have one replication site. They don't have an origin necessarily. They have multiple. But they're still going to have these replication bubbles. And inside that bubble is going to be leading strands that go in the same direction contiguously, anti-parallel to each other. And behind them, they're going to have these discontiguous strands of lagging. And this thing, because they're not circular, there needs to be a way to hold them together. So while the blue strand is going to end up with the red copy, just as the example from semi-conservative, and the black strand is going to end up with a copy that's green, semi-conservative, complementary, antiparallel, same genetic code, they are tethered together at the center by a centromere. Okay, we're going to cover in a great amount of detail DNA polymerase, leading and lagging, and the replication fork and the enzymes therein in the next lesson. And after that, we'll cover transcription. But what I want to do here is reiterate the similarities and why it's so important to keep your DNA straight, 5' left, 3' right, the strand you care about. DNA polymerase is obviously going to be for DNA, GCAT. It has replication going on, 2 strands being replicated at the same time, which means there's going to be a template strand and a replicating strand. The replicating strand is the one you care about, 5' left, 3' right. That means de facto the template strand has to be 3' left, 5' right. So because it's the replicating strand is the one you care about, replicating strand 5' left, 3' right. DNA polymerase starts on the 5' side and moves towards its own 3' end. DNA polymerase also has high fidelity. High fidelity means mistakes aren't tolerated. High fidelity is created by proofreading, which means the DNA polymerase can look behind it and say, is that nucleotide the one that's supposed to be there? Did I actually match them right? If it didn't, DNA polymerase has the ability to cut out the last one. An exonuclease, exo meaning the one on the end, has the DNA polymerase has the ability to look backwards, and if it's wrong, chomp off the last one nucleotide. It's an exonuclease because it's coming in from an unattached nucleotide. The 3' hydroxy end is open. Exonuclease, and because it's going backwards against the 5' to 3' building direction, it is a 3' to 5' exonuclease. Compare that to RNA polymerase. RNA polymerase is going to be for RNA. So GCAU, only difference. RNA polymerase is about transcription. And transcription is going to have a template strand and a transcribing strand. The transcribing strand is the one you care about, so its orientation is 5' left to 3' right. The template strand is complementary and antiparallel, so therefore must be 3' left, 5' right. But the one you care about is the transcribing strand. RNA polymerase binds to the 5' end and moves to its own 3' end. The same so far, except that we've called the strand that's being grown the replicating strand in DNA and the transcribing strand in RNA. In DNA we had a T, RNA we had a U. See how similar they are, especially if you can get the orientation correctly. And transcription also involves a coding strand, but the coding strand is the transcribing strand only as a DNA. Look, oriented 5' left, 3' right. So the only thing you have to remember is that template strand is backwards. In everything else, it's going to work. 5' left, 3' right. And RNA polymerase is low fidelity. No proofreading, no exonuclease activity. Right. So the main purpose was to say, hey, this is what semi-conservative means. Check it out in prokaryotes. Look at that origin site. Ooh, this bubble looks complicated. We'll cover it in the next lesson. But most importantly, it was used to show you that if you keep the orientation of the strand you care about, 5' left, 3' right, it'll work out such that the enzyme starts on the left and moves to the right. The strand starts on the left and moves and grows and adds on to the right. It doesn't matter if it's DNA polymerase or RNA polymerase, They're both going to do the same thing, 5' left, 3' right. The details of each one might change, but the thing that actually matters for you to figure out what's going to happen next, what's the right direction, is all about you putting the strand you care about 5' left. That is Introduction to DNA and RNA Synthesis.